Thank you, Dr. Perez. Hi, everyone. The title of my research project is Investigating the Role of NaV1.1, TRPV1, NaV1.8, TRPV1, and ASIC3 in acid-induced pain signaling in PC12 cells. And my mentor is Dr. Perez David. Ooh, sorry. First, I would like to say a big thank you to Papa Jack for supporting the Kay Fellowship and allowing me to conduct this research. Okay, so I want to first talk about chronic pain. So, chronic pain is a prevalent problem that affects millions of people worldwide, and current treatments for chronic pain are either ineffective or they rely on opioid-centered methods. So, approximately one-fourth of the adults in the United States are facing with chronic pain, and this has led to the loss of approximately $635 billion, which goes towards medical costs and also reduced work productivity because of chronic pain. So, the opioid epidemic is responsible for nearly 1 million deaths in the United States alone in the past 10 years. So, I'm sure most of you have heard of the opioid drug, which is used to inhibit chronic pain. And the issue with opioids is that it is very addictive. And when taken in large amounts, it can amount to death. And I'll be explaining later in my presentation why they are so addictive. But if you look at the graph, you can see that the rise of deaths due to opioids have increased significantly since 2013. And this number is continuing to increase in recent years. So before I talk more about the opioid and how my research aligns with that, I want to first talk about neurons. So neurons are basically cells inside our nervous system that allows us to feel pain. And there are billions of neurons that make up our nervous system, and they help us to feel pain by transmitting electrical signals. So there are approximately 86 billion neurons, but they all talk to each other by transmitting a signal through one neuron to the next. So they do this by going from the head or the dendrite to the axon or the tail. And they transmit electrical signals through a process called the action potential. So action potentials are created by ion channels inside the neurons. So essentially what ion channels are is that they are proteins that allow for um, protons to flow across the cell membrane. And this results in a difference of charge, which leads to the action potential. And action potentials are what allows the neurons to actually be able to communicate with each other. And that communication is what eventually results in the perception of pain. And there are actually more than 300 different ion channels in the human genome or the human body. But I'm only interested in three, and I'll be explaining why I chose to study those three later in my presentation. So my research question, tying with the opioid, is are there any non-addictive solutions to acid-induced pain? So first, why did I choose to focus on acid-induced pain? So acid-induced pain is triggered through an acidic environment or acidic damage to tissue, and this can lead to chronic pain if it is very significant. And essentially, it triggers specific ion channels that are involved with acid-induced pain, and this can amount to chronic pain over time. And the issue is that current treatments are not really centered on chronic pain from acid-induced pain, and also current treatments are also not very effective. So that's why I'm studying acid-induced pain. And another reason is because it is easy to mimic acid-induced pain in the cells that I'm studying because I can merely change the pH of the media to induce what acid-induced pain would be like. So now I'm going to be going back to the opioid and talking about why it is so addictive. So opioids are addictive because they're located in the central nervous system. And the central nervous system is basically a part of your nervous system that includes the brain and spinal cord. And your nervous system actually is made up of two systems that help you to perceive pain. So the opioid is, it binds to a receptor located in the central nervous system, and this causes the release of dopamine. So dopamine is basically what allows you to feel a sense of pleasure, and this can become very addictive over time. So because the opioid is involved in binding to receptors in the central nervous system, it can lead to addiction. So what I'm trying to do is I'm trying to find an alternate way to inhibit pain from acid-induced pain that is located in the peripheral nervous system. And the peripheral nervous system is everything that is excluding the brain and spinal cord. So if I'm able to find an ion channel to inhibit in the peripheral nervous system, then I can inhibit pain from acid-induced pain in a way that is not addictive because it is not tied to the brain. So NaV 1.8, TRPV1, and ASIC3 are all ion channels involved in acid-induced pain signaling that are found in the peripheral nervous system. So these are the ion channels that I chose to study in my research. Now, NaV 1.8, TRPV1, and ASIC3, they all contribute to the perception of acid-induced pain, but little is known about how they interact with each other. And also, GAPDH was chosen as a reference gene because they remain stable across various pH levels. And this is important to help me normalize my data. So more about why I chose to study these three ion channels specifically. So it is known from previous research that they are involved somewhat with acid-induced pain signaling. However, the extent to which they are involved with it is not very clear. And also, previous research hasn't been able to really investigate how these three specifically interplay with each other and interact with one another. So I'm hoping that through my research, I'm able to determine how they are related to one another and the extent to which they are influencing acid-induced pain. And yeah. So how will I be measuring this? So I'm going to be looking at something called gene expression. So gene expression is basically what happens when so basically you have DNA in your bodies, and DNA encodes for proteins. So they do this by a process called transcription, where DNA is transcribed into messenger RNA. And messenger RNA is essentially what I'm measuring. It is like the gene expression, and this is important because mRNA is later translated to proteins, and proteins give rise to ion channels. So essentially what I'm doing in my research is I'm detecting the amounts of mRNA that are present in my experimental groups, and this leads me to determine how much of ion channels are present because of the amount of mRNA that is present. So more mRNA or higher gene expression equates to more proteins that encode for ion channels. And the more proteins there are that encode for ion channels leads to a greater sense of pain from acid-induced pain. So I worked with PC12 cells in my experiment, and PC12 cells are cells that are derived from a rat tumor. Now, this might sound a little bit concerning, but it is actually good that I'm using cells from a rat tumor because the tumor allows there to be lots of cells. And it's also totally fine that they're from a tumor because they act just in a way that is very similar to how a human neuron acts. So the tumor doesn't have any negative impacts on the cell function itself. And also, I'm using PC12 cells because they express all the ion channels that I'm interested in, which are again NaV 1.8, TRPV1, and ASIC3. So here is my procedure. So this is a flowchart that illustrates what I did in my research. So the first step was cell culture of the PC12 cells. Then I exposed two groups to separate pH levels. So the experimental group was exposed to a pH of 5.6, and the control group was exposed to a pH of 7.4. So the pH of 5.6 is essentially to stimulate what an acid-induced environment would look like that would lead to acid-induced pain. And 7.4 was just a control. So after I did that, I extracted the mRNA from 4 different time points for each of the samples. So I chose time points of 30 minutes, 1 hour, 6 hours, and 24 hours. After I did that, I converted the RNA to cDNA. Now, the reason why I did this is because I'm using qPCR, and qPCR it works better with cDNA because cDNA is more stable than RNA, and it is also easier to replicate cDNA than it is to replicate RNA. And after I converted RNA to cDNA, I put all my samples inside the qPCR, and then I did data analysis. And this leads me to my results. So essentially, this is a graph of the gene expression being the y-axis and the time points being the x-axis of NaV 1.8. So what I found is that NaV 1.8 expression under acidic conditions peaks at 6 hours. So as you can see, there is a large difference in gene expression levels at 6 hours for a low pH and a normal pH. And For TRPV1, what I found is that the expression peaks at 24 hours. And as you can see in the graph, there is a large difference in gene expression for the lower pH than normal pH. And as for ASIC3, the expression remained relatively consistent with the control. And although the expression for the cells grown in acidic environment were slightly higher than the control, it was really consistent and very close. So what do these results mean? So based on what I learned from this, I was able to conclude that NaV1.8 and TRPV1 are likely key players in chronic pain from acid-induced pain. Now, why did I conclude this? Because NaV1.8 and TRPV1 both showed a large difference in gene expression when exposed to an acidic environment versus the control. So this leads to a larger difference in the amount of ion channels that are produced when these ion channels are exposed to a lower pH. And the more ion channels that are produced, that means that there is a greater perception of pain. So it is likely that these two ion channels are related to chronic pain from acid-induced pain. And on the other hand, because ASIC3 remains relatively consistent with the control, it probably means that it is most likely a baseline acid detector. So it does have a role in acid-induced pain signaling, but it probably doesn't lead to chronic pain like NaV1.8 and TRPV1 do. So future research should look at protein activity and drug inhibition of NaV1.8 and TRPV1 to treat chronic pain that arises from acid-induced pain. And also during my honors independent research project, I'm going to be using RNA sequencing, and this basically allows me to look at other ion channels that are involved with acid-induced pain signaling, and it also can reveal what pain signaling pathways are involved. So it just helps me get a better insight of how this whole process works and what contributes specifically to chronic pain that arises from acid-induced pain. And so this research has definitely taught me a lot of things, but most importantly, it has taught me how to be responsible and to not procrastinate. And it is really important to really understand what you are doing while you are researching and doing your experiment. And it is also important to read your literature and do everything ahead of time so you don't feel stressed out. And most importantly, this research has taught me to remain calm even when a mistake has been made. So when I was extracting RNA to turn into cDNA, I accidentally threw away RNA from one of the samples. And even though I made like a huge mistake like that, I just decided to remain calm and I just kept conducting my experiment to make sure that I wouldn't repeat the same mistake again. And thankfully that didn't happen again. So my data wasn't like completely screwed. And also, just some advice to future researchers. Don't be afraid to try new things and always ask questions. And like the mentors will never like ding you for asking too many questions because it's important for you to know like what you are doing and to make sure that you are doing things right so you don't make silly mistakes. And also, if you are interested in doing research, I would highly recommend applying for the K Fellowship because it allows you to do so many things that you want, and you have people here to support you, and it is a very, very rewarding process. And I also want to thank Dr. Perez significantly for all the help he has given to me, and for Dr. Chan, Mrs. Kobayashi, and Mr. Hill, who helped me with qPCR, and all the faculty who helps me with my research. And here are my works cited. Thank you.